This is the final session, and I'm delighted that so many people are here, and I think it just shows really how very important this topic of dosing and redosing really is to our sector. And it's been the topic in our sector ever since I've been in it, 30 years now. And I still think we're very much trying to find the answers, and I'm sure the panel today, with their expertise, will really contribute significantly to our discussion. I'm happy to take questions. The lights are so bright, so I can't see your hands if they go up, but I'll rely on one of the ARM team to sort of come and just tell me, and we will take questions as we go along for our panel. So we have a really great panel today. Unfortunately, Dave Lennon had a family emergency he needed to attend to. I'm Chris Mason. I'm chairing the session instead. We have a great panel. I'm going to introduce each one, just ask them to say a few words about themselves and about what they do, their company or their academic work. So the first person is Eric Crombez, who is Chief Medical Officer at Ultragenyx. Eric, would you just like to say a few words about yourself and your company?
Great, thank you. Eric Crombez, Chief Medical Officer for Ultragenyx Gene Therapy. I've been working on our lead gene therapy programs for the past 7 years, biochemical geneticist by training, so really spending the entirety of my career in the development of new treatments for rare genetic disorders.
Fantastic, thanks Eric. Our next person is Peter Francis, who is CSO at 4D Molecular Therapeutics. Peter.
Thank you very much, a pleasure to be here. Yes, Peter Francis, I'm an MD, Ph.D. clinician scientist. I'm the Chief Scientific Officer at 4D Molecular Therapeutics. I've been with the company for several years, in total working in the biotech industry in AAV gene therapy for about 15 years, previously in academia, trained in the United Kingdom. Pleasure to be here.
And next is Tim Miller, who is CEO and President of Forge.
Hi, thanks everyone for coming to the last panel. Tim Miller, President and CEO of Forge, about 20 years in gene therapy, going back into the 90s, and you have built out a variety of different AAV programs and have focused on the way to do some redosing for a number of them. And if you don't like the answers, I'm sure that there is a gong that if you hit it, okay, one of us will slide into a pool of water. At least that's what it feels like up here. So
I've got it here, Tim. It's just in my hand. Right, okay.
Somebody had one.
Yeah, no problems. Finally, Nicole Paul, who is Professor of AAV Gene Therapy at the University of California at San Francisco.
So yes, happy to be here. So Nicole Paul, my lab at UCSF is fairly unique. So we try to kind of take a 10,000-foot platform view of the field, and we're really agnostic to any particular species, organ, tissue, cell type, any of these types of things, and try to develop enabling technologies for the entire field at kind of any aspect of the AAV development pipeline when it comes to just making AAV gene therapies kind of cheaper, easier, and faster for everyone to use.
So I'm probably going to start out with the hardest question, okay, guys? And that is, what is a dose? What exactly is it? Is it just the number of viral particles? Is it something to do with the potency? What's it all about? Nicole, I can see you're very keen to start that one.
Sure, absolutely. So I think that depends on who you ask. So as the academic on the panel, I'll answer in this way. So certainly when it comes to what we need to report, it's absolutely just the vector genome, the vector genomes per vial that you're going to be administering to the patient based on most often per kg body weight. But in reality, and I think the reason that we have this panel here today is that that's probably not all that needs to be reported and not all that needs to be discussed and thought about. And so starting, certainly moving forward, I think after kind of the recent events of the last year, thinking about the importance of what else is in that vial, whether that's empties or other impurities, and the importance that these may be having as it pertains to both potencies, but also potentially safety and immunogenicity effects. So I think moving forward, we won't just be reporting, what's the vector genomes per kg that we're all giving to our patients, but probably a number of other metrics.
Eric, how do you see it?
I guess with the list you started with, I would say yes to all of the above. I agree there's a lot of ways you can come at that answer, and I do think when you think about how you're dosing on genome copies versus full empty ratios versus the quality of the product, I guess for me, importantly, as we pull these gene therapy programs through the development process, to me, dose or effective dose is really, what delivers the most therapeutic benefit to patients with the best safety margin.
Yeah, safety is obviously critical. Dose obviously has a big impact on efficacy, safety, manufacturability, cost of goods. It goes to just about everything. So I think this is such an important topic. Tim, what's your view on this?
Yeah, so I think certainly with the FDA AdCom recently, there's definitely the focus on the empty full, right? Because when we think about dose and we think about VGs per kg, for example, it's it's the fulls that we think about. But there's such a wide variety, I think, in the amount of empties that many of us go through our manufacturing process. But I guess I'd probably take the question really just to one more level. It says it's not so much the dose that we administer, but it's the dose that actually gets into each organ. Right, and so taking it more to the organ level about what really is going to be an efficacious dose as we think about the liver or the brain or the kidneys. I think Chris was saying on one of the panels earlier. So just that's I think how we're starting to think more down to that level when we think about dosing.
So Tim, just to follow up on that, I mean, are you thinking we should be dosing more, say, instead of per unit body weight, more sort of unit liver size, for example?
Yeah, I think that's probably where some of us are going to go. I mean, you see a number of studies that are looking at, they dose by brain volume, right, for example, now. And so, it's easy to look at a dosing on a systemic level, but, as you think about some diseases that have, enlarged livers, so do you have to take that into consideration when, you're actually doing that dosing because it's going to absorb more?
Peter, you're in a rather advantageous position. You've got the eye. When you think about dosing in the eye, how do you think about that?
Yes, I was going to say that the conversation up to this point is really, I think, focused on IV delivery, but compartmentalized delivery is a whole different calculation. If we just focus on the eye for a second, then dosing per kilogram is not typically what we do. It'll be a total dose vector genomes. But I think the same principles apply. I think the field has now set itself a challenge because clearly high dosing is associated with more complications. So it's incumbent upon us to develop, I think, new vectors that can reduce that dosing. And then to Tim's point, it's not all just about the capsid dose, it's about the amount of expression you then get in the tissues. And so I think it's incumbent upon us to look for ways to reduce that dose by having more efficient gene expression.
Yeah, I absolutely agree. Nicole, sort of on the more sort of controversial side, I mean, it has been suggested that, if you do get some waning of AAV efficacy, perhaps the idea might be to give a larger dose at the beginning so that over a patient's lifetime they would still have above the threshold level. What do you think of that view?
I think that depends on whether or not you're administering systemically or not, what the target tissue is, what the capsid is, what the payload is. So every one of our programs, I say ours if I have a program, but every one of our programs is a bit of a snowflake. So I think there's probably no one straight answer that would apply to everybody. I get nervous when we talk about, let's just give a higher dose and hope that it lasts longer. That makes my skin tingle. I think we do need to be cognizant of the risks. Even if the last year hadn't happened, I think we need to be cognizant of the risks of let's just increase the dose. I think we should all, put our academic hats on in that sense and think about ways to make these more efficient and more efficacious and more potent so that we don't need to kind of use really blunt hammers and just increase the dose without having good reason. And I would rather, if I put my patient hat on, I would rather receive a dose that will wane 20 years from now, but I don't have huge safety risks up front, and wait for the possibility of either new technologies or ways to redose, and we can certainly talk about that in this panel, and get redosed in 20 years.
Tim, what do you think? I mean, it obviously hinges upon a little bit on redosing, which I want to talk about later, but giving a larger dose at again sustained level over a lifetime, given you're going to maybe lose some efficacy.
Yeah, Chris, I think that I think the field's moving not so much towards a higher dose as much as it really is towards an immune modulation strategy that's going to extend, that dose. So again, as we think about it, and we'll probably talk a little bit about this, but if you go through a significant immune suppression or even an ablation step, there's ways that you actually increase the infectivity period, right? So if you can increase the infectivity period, you can actually do this at a lower dose. You might have some additional safety. So there's a couple different ways that we think about that from really the immune modulation.
Eric, do you have any comments to add to Nicole and Tim?
Yeah, and I think absolutely agree. And I think with the idea that you do want to hit as many of your target cells as possible, we have been very on targeting liver-directed gene therapies, but certainly moving on to the muscle space, moving into the central nervous system. And as we do so and think about higher doses, I don't think that the concept of more is better is the right way to think about this. I do think there's a limit there, and we need to move through our doses and do so safely. So I think, as we move forward as a field, really talking about tropism, how we're going to better target, how we're going to handle any immune response that occurs, and really kind of build upon the work we've done so far.
Just to continue on that, I mean, do you think that dose is only about, to do with the actual particles or vector particles, but do you think maybe speed of infusion plays a role in that? If you put it in, I can see Tim nodding now. So basically, you give a big bolus, and maybe the early cells get a huge amount of the vector going, and the ones downstream get a little bit less. Do you want to comment on that first, Peter, or Tim? I don't mind who comments.
Tim is raring to go.
Well, I had a
hand the floor to him.
He jumped.
Well, there's another way that we also think about this, right? It's that, when many of us are trying to administer these gene therapies, we think, we're going to go intrathecal, we're going to go IV. Well, I think what the past couple of years have seen is multiple routes of administration at the same time, right? So you're starting to see programs that use an ICV and an IV or an IV and intrathecal. Why not do more, right? I think we were developing a drug at one point, an AV therapy that had an IV and an intrathecal administration, but those kids were still going to go blind. So, the field, I think, is moving to this point where you can actually lower maybe some of the systemic dosing, but you can actually go to a point where you can do more routes of administration to be able to get more really shots on goal.
Nicole, I can see you nodding. Is that?
So I think the two things here, in combination with multiple routes of administration within a single program for a single patient, but also the implementation and kind of the add-on technology of using either new capsids or new regulatory elements that will also be multi-tissue. I think we've all been laser focused, whether in the academic space or in a number of the companies that are doing engineered capsids, on getting capsids that go to one tissue at the expense of all others. But I think the next generation of these will be combinatorial. I'd like something that goes to both the heart and the skeletal muscle, but not liver and not anything else. And so starting to be able to get the multi-tissue indications where we can use a single capsid that can go to all of the different tissues that we're interested in, also regulatory elements, promoters and enhancers and these types of things that can together help us get this tissue-specific at a multi-tissue level granularity in combination with better multiple methods of administration so that we can hit everything, and that will be kind of the next
wave.
I
mean, just to continue on from that, I mean, the challenge as I see it is that you still end up with some cells receiving many copies of the AAV and some getting very few. And I think the liver, 1 or 2% get the cells, get the AAV typically? I mean, is that part of the secret of dosing, getting somehow better distribution throughout the target tissue? In this case, how would one go about it?
Was that to me?
Yeah.
Yeah, we certainly see, and some of this is capsid specific, and some of this will be like dose, volume, pressure specific on how you're doing your infusions and all of these things. But there are certainly hepatocytes. We've seen this most notably in a number of the hemophilia programs, where there are the super transduced hepatocytes that get thousands of copies, tens of thousands of copies of your AAV, and many other nearby ones that don't, as you're going along that acinar axis for either periportal or pericentral hepatocytes for the niche folks in the audience. And I think that there is a lot that can be done in both the capsid and regulatory element space, but also the methods of delivery, even within systemic. We're very often administering these things either at the hand or at the elbow, but there are other entry points, and do those have differences when it comes to getting different cells transduced along that acinar plexus in the liver? And I think there's a lot more research that could be done there, even within intravenous administration, to find new entry points and how that might affect things.
Peter, this is a challenge in the eye, isn't it, as opposed to where you actually put it in. Do you want to just tell us a little bit about the eye?
For sure, yes. The eye has proven an excellent place to go with gene therapy. There are several amenable routes to delivering your AAV vectors, which have been shown to be beneficial from a therapeutic perspective. One of the methods is a very involved surgical approach, which is subretinal delivery, which allows you to deliver a vector directly to the target cells, but in a relatively small and circumscribed area, and the AAV gene therapy typically doesn't spread beyond the area of the treatment. Other approaches are now being taken where you can give intravitreal, and so directly into the cavity of the eye, your gene therapy, and then that diffuses and distributes to the retina, in this case, more broadly. In those instances, there are some other considerations. And most recently, there have been attempts to and demonstration of potential efficacy by suprachoroidal route, where rather than penetrating the whole surface, all of the coats of the eye and delivering into the vitreous cavity, in this case there is a suprachoroidal space that sits between the retina and the sclera, and so you just need to penetrate the scleral space through the sclera in order to deliver your agent. And each of those is showing has shown benefit. The best approach is really limited by the type of vector you're delivering, the efficiency of tropism, which cells you're going after, and what disease stage. So, for example, subretinal delivery would be more targeted towards those with residual areas of vision, more advanced cases perhaps. If you wanted to target earlier cases, then an intravitreal approach or a suprachoroidal approach might be most applicable.
But I guess part of the equation also, when we think about dosing and actually treating patients with eye disease, is that eye disease is very common. Or many of them are very common, and if you really wanted to make a big dent in the population, you'd need something that was very simple, quick procedure, ideally in the high street. And does that play into your thinking when you sort of formulate your therapies?
I think that's a really good point. There are some very large market indications for which gene therapy is suitable in the eye, including age-related macular degeneration, both the wet and the dry type. And there you're absolutely right. For that particular population who are often elderly, an involved surgical procedure potentially to both eyes would be disadvantageous over an office procedure like intravitreal or suprachoroidal delivery. So that's a really good point. If you wanted to commercialize to a very larger population, then that quick and easy, safe, office-based approach, maybe not the high street, at least in the United States, but certainly the office-based approach would be very, very suitable for that population, who in many cases are quite used to receiving, for example, intravitreal injections on a monthly or bi-monthly basis.
Eric, has your company looked at different delivery routes? I mean, is that something which you consider as a priority?
The delivery routes?
Yeah, delivery routes.
Yeah, so we have been very focused on peripherally administered gene therapy, and our original 2 programs were in hemophilia, and certainly we learned a lot there. And I think, to the earlier point, when you're looking at excreted proteins, it's very different because you can make the argument you need to target a fewer number of liver cells to really producing that factor, but then you also take into stress on those hepatocytes and things like that. And then with our other inborn areas of metabolism, really focused on trying to target as many hepatocytes as possible because we really need those enzymatic processes, those metabolic pathways functioning as many hepatocytes as possible. We do think with some of these targets, we've selected there is a sink effect and you can compensate for other hepatocytes you have in target, but that's important there and different from hemophilia. But as we've moved into muscle where you need much higher doses and now with the central nervous system, yes, I think how we deliver that gene therapy has become much more important to us.
I'd like just to switch just a little bit. I mean, a lot of the dosing thing actually comes down to manufacturing as well, consistency. And I think the panel's really nicely mentioned the fact that empty fulls is a common issue. Are other factors like, say, the capsid? I mean, AAV's got its 3 different proteins in the capsid, and we always say the ratio is 1 to 1 to 10, but that's Nicole, I can see you smiling. That's just not the case. And does that make a big impact, that the fact that there is this variability in the capsid that could potentially lead to change in efficacy or maybe change in safety? And can we control it? Can we control those proteins in any way?
Can we control it? I mean, the scientist in me says anything is possible. So yes. Are we controlling it right now? I don't know. I think we're still kind of in the early days of using some of the earliest technologies for manufacturing AAV. But in many ways, I don't know if I'm the best to answer this. This is probably going to be best answered by Tim, our CDMO guy on the panel.
Well, I think that there's an element of truth there. I mean, it's much easier to make it, a single-stranded AAV9 than it is maybe a self-complementary AAV2, for example, right? So, so every program I think that we've seen is a bit bespoke, right? And so when you start thinking about, the novel capsids that more and more groups are trying to push forward and just looking at some of the variable loops, I mean, sometimes a single amino acid change can make a huge step in your manufacturing or manufacturability. So, I think there is an element of that, Chris.
So more to do on the CMC front, but it's early days. That would be the conclusion. I think a question that I know everyone's thinking about is the recent FDA Advisory Committee meeting. and its impact on dosing. I know they said they weren't going to set levels, but what's your views on this? It'd be really great to go along the panel. Eric, do you want to kick off?
Yeah, so I have the good fortune of being one of 2 industry representatives sitting on that advisory committee and participated in that 2-day panel heading into Labor Day. And I think going in with the idea of focusing on safety, and that was originally the original idea put out there, was interesting, and I was glad to see that as we prepared for the meeting and then had the discussion, that it did shift very much to benefit risk, and I think that really is a discussion that has to take place there. I think the briefing book was interesting. It laid out a lot of questions that we've been really trying to wrestle with for a very long time, and some we've been thinking about for a long time, some that are relatively new. I was happy with the way the conversations went, and some of our discussions we've been having in follow-up, with really coming back to these programs as individual programs. There are very important key differences with these different types of programs, and while we can think about and learn about the platform as a whole, we need to come back to these programs as individual programs and really treat them as such.
Understood. Peter?
Yes, thank you. I wasn't privileged. I was an attendee. And I think it was very timely, given, as Nicole said, what we've been through over the last year in gene therapy, and it really is a focus now on safety. And I think a lot of the points have been made by the panelists here today about lower doses and how to achieve tropism and satisfactory transduction and long-term expression with lower doses. And I think the point is well made that this is very much on a disease-by-disease basis as well. So I think it was a rallying call, I think, to the industry and to the academics that we do need better vectors with better tropism. We do need to explore tissue-specific promoters to reduce off-target expression. And then we also need to look at our overall payload design to avoid issues such as immune activation, ADA development, and ultimately silencing if we're going to get long-term expression. So, I think there's important take-homes from that meeting.
Tim? I'm sure you watched it very keenly.
Yeah, we had a big conference room when we sat with a bunch of popcorn. But no, it was important. I mean, the FDA has to shine a light sometimes into the dark corners, right, for the issues of safety. But I think that I don't think that anything came out of that was necessarily a surprise, and I don't think that anything came out of that was something that was going to plant a flag in the ground that said, now we've got to really, 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 be worried about something. I do think it helped raise a lot of the awareness and made a lot of us push, probably the empty full question a bit more. But it also helped settle a lot of the issues, things like integration. Never been shown before ever in humans, right? So type of thing. So risk was very, very low. But I think it was helpful overall, and I think that the field probably, we've moved on very fast, right, from that.
Yeah. Nicole?
Yeah, I was surprised, given all of the companies that I either consult for or SAB for, or just casual conversations that I've had with colleagues, the pent-up angst around that meeting, I think, was pretty high, in some ways unjustified. I don't think the FDA often comes down particularly hard on these types of things, especially given the data that we're all aware of that's publicly available. So I was pleasantly surprised and also not surprised at all that it was fairly uneventful. I don't think that they were going to set an upper limit on dose, which I think was the main thing folks were very secretly cringing about, is were they going to set an upper dose at this meeting? And I don't even know if that would be possible given the differences that we all have in how we titer vectors. One company's 1E13 VG per kg is not the same as another company's 1E13 VG per kg. So given those challenges with the methodology that we're using around titering, which is perhaps a separate conversation, I don't think there's going to be an upper limit. I think all that we could potentially expect to really see as far as new guidance or new rules or new suggestions coming from the FDA might be around the empty full, not in, again, setting a limit, but perhaps asking folks to start reporting that. And then also perhaps a little bit more around the requests for longer follow-up with things like liver ultrasounds and these types of metrics for intravenous infusions.
Yeah, Nicole, you raised a question I was going to talk about, but the whole thing about this is, how good can we actually assay the actual drug product? And you raised an important one about titering, which is very fundamental and yet really hard. And we all end up with different numbers because we all do it different ways. I mean, is it now time that there should be a standard for actually getting a titer so we can compare company A's with company B's doses?
Well, what's a standard? That's a separate question. So we have this in a separate thing I'm a part of. So the Somatic Cell Genome Engineering Consortium, we've been trying to develop standards for AV in collaboration with NIST, and even just deciding what that means is a contentious issue, and I don't think we even have an answer to that, because even if you made an AAV8 and AAV9, which we have had, AAV2 and AAV8 now available for purchase for a while now, someone's single-stranded AAV2 is not the same as someone's self-complementary AAV2. And, two groups that have, different payloads with different secondary structures and all these types of things, they're very much not comparable. So I think it might be a challenge in order to come up with what is a standard everyone would even agree on. So making one available and requiring folks to use it, I don't know that it's possible today yet.
But it is a challenge. I mean, investors do look at the numbers. They go, ooh, 10 to the 14, bad. I mean, I see Peter nodding. I mean, that's correct, isn't it, Peter? There is this element of see a number and they believe it because they must all be the same. And we all know on this panel, and everyone in the room knows that 10 to the 14 in one is maybe 10 to the 13 in another. And that is actually causing us, I think, challenges. And so maybe we do need to think a little bit more about it. Tim, how are we going to fix that?
Well, a couple of years ago, I think the FDA moved the goalposts on all of us when they started to ask for % CVs, right, when measuring titer. And it's forced, the entire field really to move towards a ddPCR. Yep. So, or, somebody with very, very steady hands who doesn't drink coffee a lot, but I think that's helped us, as a field, and at least it's going to provide more consistency, lot to lot to lot, under the same program at usually the same manufacturer, so.
And even empty capsids, Peter, this is a common problem, the ratios. Most people seem to accept a range on their empty capsids. How do you see that as playing into this?
Well, I think, to Nicole's point, we're in the earlier stages of manufacturing. There's a lot of enhancements that need to come, and I think this is a driver. I think that there may have been days gone by where there was an acceptable amount of empty capsids, but I think those days are over, and we need to minimize those and continue to minimize those. I think the industry has moved to minimize empty capsids as best it can, but clearly there's a long way to go. And I think now what's emerging in terms of the safety risk for higher doses of overall capsid, it really is incumbent upon us to absolutely minimize the amount of empty capsids that we see in our particular products.
The challenge here is we use this word empty capsid, and it sounds nice. They're empty, they don't contain anything, but of course they do. Mm-hmm. And that's the challenge, isn't it? They do contain material from the 293s and other stuff, which you really don't want in the patient. Eric, when you think about this, your company thinks about it, these empty capsids, do you have set a very tight specification on what you would expect accept as a drug product?
Yeah, I think, with everything, there has been a lot of evolution and a lot of innovations that's been coming through, and a lot of people have been working on this for decades. So I think, there's the programs we're moving into our pivotal trials now, and where we are with those programs, and really trying to drive to the best ratio we can, having the most full capsids available. But then as we build out our pipeline and bring more programs along, particularly again as you move into bigger indications that may require higher doses, then I think you really do need to focus on continued innovation and really controlling what you control, but accepting that it takes time, and that innovation is important, and it's important to build upon.
Yeah, Eric, just to stick with you, when you think about dosing, do you use animal studies, or what do you use to actually sort of work out, or what's your first dose going to be in patients?
Yeah, and I think, with that prevailing view, that I was happy when I first started working in the field with the FDA really accepting that you don't need to start with a pure safety dose, because with this idea that it's one and done, and I guess that gets to the conversation on redosing, that it's important to really start with a dose you think may be or will hope to have some level of efficacy, but then again, that comes maybe with some potential, with some safety concerns there. So I think that was a great place to start from. That was also very important to us as we built out our initial pipeline and focusing on the liver, because we thought we could learn and really pull through those learnings through our pipeline, so we understood really what, in our hands at least, what were a safe dosing range again, we were very much in the E12, E13 range, but then we're moving into E14, and obviously that becomes a point where people want to have a lot of conversations on potential safety risks.
Yeah, I mean, that is our big challenge, isn't it, ethically, to go in with a dose you know hasn't got any chance of working, but would be a good safety dose, you can't do. And so we are trying to approximate, well, what would start to give that efficacy that would benefit the patient long term? How do you think about this, Peter, in the eye?
In the eye, I mean, I agree exactly with what Eric has been saying. We have a large number of animal models of eye diseases. So you can, if you like, set your entry dose with those. Unfortunately, they're mostly in mouse and rat. And so the extrapolation to a human equivalent dose is very challenging. And as you rightly say, we're incumbent upon us to go into patients with the disease with something that is potentially therapeutically active. I think there has developed over recent years a segue and translational ability from the non-human primate, where the eye itself has a structure that's very similar to the human eye, the human primate, and the size considerations are much similar. And I think that sort of translation from mouse models into non-human primate testing, albeit we don't have many non-human primate retinal disease models, but just even the normal primate eye can really lead you towards that potentially therapeutic dose, and obviously gives you a very nice safety range as well in an eye that approximates very much to that of the human.
Tim, what do you think? I mean, we as a sector are doing pretty well. We had the figure the other day of 3,500 patients treated with AAV. That we've got a pretty good track record. I mean, but this dosing is complex. And how do you assess initial dose so it's safe but with potential for efficacy in the patient?
Yeah, it still harkens back to some of the empty full question, right, that you had a few minutes ago. And as part of the evolution of this field, you're moving to new cell lines, right, for example. So we just put out one of our cell lines, up to 50% fulls, coming out of that improvement over the industry where it would have been 10 or 20% in the beginning. It also has to do with how we measure, right? So if you're measuring by EM versus AUC, you're going to get probably two different results. Which is the one that's going to be more accurate? Again, the field's moving more towards that. So, but all this functions back to how we think about efficacy and what's going to be an efficacious dose. I mean, I think Eric hit the nail on the head. That you still have to give something that we think is going to work, right? So, and we actually have that mandate. Per all the development that we do, we have to be able to one and done get some level of benefit.
And Nicole, how do you think about it? I mean, I know you do a lot of animal work. I mean, mouse and I assume non-human primate as well. I mean, first of all, do you get a good correlation between, say, mouse and non-human primate, or is that even quite an art to get a correlation there?
We certainly don't see one. But I'll take a different tack to answer this question. So one of the things that we do in my lab is really try to apply kind of broad omics to AAV gene therapy, so things like proteomics and genomics. And so we try to get samples, both from other academic labs, from some of the research grade vector producers that are supplying all of the various academics and many of the startups with some of their early research grade products. And we're applying proteomic and genomic approaches to look at these and see, is what they're saying they have what they actually have? And I can almost tell you universally without selling anyone out, including my own lab, what you think you have is almost not certainly what you have when it comes to, what is the composition of this capsid? What are the impurities? What are the empty fulls? What is the genome you think you have? What are the ITRs that you think you have? All of these things are very much different in many of these cases. And so I think the variability within a single production, obviously we aren't looking at GMP preps in the lab, we can't afford that. But even with, I would argue in some cases GLP-like vectors from a number of these suppliers, there's quite a bit of variability. And this is all of that early data that everyone is using in order to determine whether or not we want to spin out this company and whether or not we want to invest in this, for this particular audience. And so a lot of that data sometimes I think is dicey at best.
Let's just change tack. I mean, I think one of the topics that frequently comes up is antibodies against the capsid, pre-existing antibodies. And how do you see that? I mean, do you want to start with this one, Tim? I mean, what do you dose a patient with who say, hasn't got the antibodies compared to one who say got a pretty high titer?
Yeah, it's the worst thing that after a parent gets a diagnosis that they can be told is that their child also has antibodies, right? So we've all had to live through that. So I think, what we're all trying to figure out is that what type of immune suppression strategy is going to end up being the one that we can give to those patients that have. I mean, the field's moved from, thinking about what was originally a total, titer to more that's more specific to neutralizing titers. And I mean, I think it's been shown that even a very small amount of neutralizing titers can really significantly impact efficacious dosing. So, what we're all trying to figure out right now in non-human primates and is, what can we do from an immune suppression strategy to reduce, or remove antibodies?
And how, Tim, just to stick with you just for a moment, I mean, how good are we at assessing the titer of the antibodies in the patients?
I think we've done this, I think, for a couple of different clinical trials where, you have to go and you have to work with a group or develop it yourself to come up with both a total and a neutralizing antibody assay. So I think that the field, I think, has it pretty well in hand at this point. I mean, both of you, I'm sure, have, the same type of, assessments. But, again, the question is, well, what do you really do after you have that information?
So, Peter, I mean, for the eye, is the antibody issue a concern, or is it sort of like the brain, it's sort of immunoprivileged?
Well, there's a lot said about the eye being immune privileged, but it clearly has its own very complex immune system. And often the systemic circulation or the systemic immune system can be involved. So I think there is some relative immune privilege. I think it's a matter of still debate whether neutralizing antibodies in the eye itself play a significant role in reducing efficacy at this time, from a primary dosing perspective. And yet again, we don't have a lot of evidence from the potential for redosing. I think that obviously contrasts significantly with other more broader delivery systems like intravenous and systemic delivery, where there's good evidence that neutralizing antibodies play a significant role. I think to build on the points that have been made, I think we need to understand more and more about what we mean by a specific titer of neutralizing antibody, which then excludes patients. And then on the other end, are we able to develop novel vectors that can evade pre-existing neutralizing antibodies, and how successful can they be? Clearly, these vectors typically will engage in the immune system, and so redosing might be a challenge. But I think there's a real opportunity for us to develop vectors which evade pre-existing antibodies due to exposure to naturally occurring serotypes for systemic delivery.
So Peter, when you think about the eye, do you have to treat both eyes at the same time, or can you treat one and then treat one later, or do you risk antibodies or a challenge when treating the second eye?
It may be dependent on the exact route of delivery. I mean, we have an approved gene therapy in Lux-Turner for LCA2 RP65 mutations, which I think has been marketed for the last 2 to 3 years, and bilateral treatment is given, at least staged, due to the surgery some period apart. And the results from those studies, from that clinical experience, has been good. So it does paint the picture that we may be able to dose both eyes separately. To your earlier point about office-based procedures, it could be also conceived that for simpler methods of delivery, such as intravitreal or suprachoroidal, you could advantageously treat both eyes in a clinical setting at the same sitting, or at least very, very close, without the need to go to the ER. Clearly, that would be potentially advantageous if there is an immune system, if there is an immunity and sensitization to the initial dose. But things look fairly positive at the moment, I think, in the eye.
That's great. Eric, do you want to take this one with the antibodies? I mean, what's your position on this?
Yeah, I mean, I think we've been talking and thinking about titer levels and the importance of them. I mean, to me, we're really, at least at this point, looking at patients who have been exposed to the native form of these vectors and those who haven't. And I think obviously we're very focused on right now on those that have not. But I think, for us it's very important, particularly in the rare disease community, and I appreciate that gene therapy is moving beyond that, but we make a commitment to these patients and to these patient groups. And I think, as we touched on earlier, you can't really just leave one subset of them behind. We really want to treat, all available patients who can benefit from this. So, I think, to me, in some sense, and I don't want to oversimplify it, but, those patients with pre-existing neutralizing antibodies become very similar to those patients who need to be redosed. So I think, as we look forward and really look at ways of doing that, I think that becomes an important part of what we're going to do next.
Nicole, what are our options from an academic perspective for dealing with this antibody issue, this pre-existing antibodies? What have we got available to us?
So there's the short-term answer and the long-term answer. In the short term, for folks who have existing programs, we've really just got to look at the currently available, immunosuppression regimens that are out there and find for your specific program what combination of 1, 2, or 3 drugs is going to be the one that's going to pair best with your patients and their age and their specific, kind of disease needs for your particular program. In the longer term, as we look forward to folks who are thinking about, instigating a new program and who haven't started it yet, there's much more kind of opportunities available. And without revealing anything that I've read when it comes to grant reviews and upcoming papers and abstracts and all kinds of fun things. There's some fun things coming up the pipe that folks are working on throughout the space, both industry and academia. So it's certainly a focus area, and there's going to be some new things for folks to play with coming forward. But again, those are going to need time to get sussed out and see how well they could work in the clinic.
So, that takes us nicely to this whole question about redosing. Tim, we'll start with you. Tim, first of all, some indications, maybe they are once and done, but others may well need redosing. So what's your views on, how are we going to go about ones where redosing is going to be required, say, for a regenerating organ where you've got some degree of washout?
Yeah, so kind of to draft off of what Nicole was saying, there's the long-term question and there's the short answer. I think that right now, for a lot of the diseases that we're trying to evaluate, past 5 years has seen the field move, and I think accept the terminology of treating earlier, treating with more is better. And as we see more and more newborn screening, the question will actually probably come more into the social conscience, right? So that because you're going to get patients that'll be treated, under 1 year of age, and because AAV doesn't stick around for more than 2 decades, let's just throw a big number out there. What happens when they do need something like that? So, what can you do at the beginning to help with that? And we actually have a program, first of its kind in the world, that uses a immune ablation step, to give a bone marrow transplant. So you solve for a lot of the systemic issues that you might see. At the same point, you're removing the immune system. But Chris, to the point really you're trying to get at, what that really ends up doing is it creates a much longer infectivity period. So that 3 to the 13th isn't 3 to the 13th anymore. It's like 5e to the 14th. Because there's no immune system, you get a longer period. Those kids are probably not going to need that level of redosing for a much longer time frame. So I think there's strategies that are out there that can be used, but again, it's how do you treat a 10-year-old, right, with something, and a few years later they're going to need to be redosed. I'm not sure we have that answer yet, but I'm looking forward to seeing some of those grants come out, and we'll see.
What do you see about the eye? Do you think the eye is one where single dose and we're done, or do you think that's likely an area redosing may be required as well?
Well, I think it'll probably be disease-specific and retinal disease-specific, but we must entertain the possibility that redosing may be necessary. I think it's likely, we've demonstrated from preclinical studies in multiple models that you can get long-term efficacy from a single dose. To your earlier point about treating the other eye, that's almost, in some ways, from an immune perspective, could be like a redosing strategy. However, I think we need to entertain the possibility that redosing may be important. I think points have been very well made about the structural changes in growth and so on. The eye, obviously, from a fairly early age is the adult size, so we don't have those kind of considerations. One possibility from a redosing perspective is that we might have enhanced efficacy. That might be more applicable, for example, to large market indications where you want to create long-term anti-VEGF, for example, for diabetic macular edema treatment, and you might wish to redose to improve the efficacy in a disease that is otherwise progressing. I think also there's a potentially unique environment in the eye as well, given we have approved therapies and a number that are showing progress, is that, as I alluded to earlier, there are different routes of administration. It's quite possible that we may face patients who had successful subretinal delivery of a gene therapy who now might benefit from additional suprachoroidal or intravitreal gene therapy to get enhanced efficacy. So we need to think about how those challenges will be overcome to maximize the benefit to our individual patients.
Eric, just still on the redosing, but dose is a problem, how you define it. What do you think about? You might put in a small amount and then dose again to sort of tighter up to the clinical efficacy you want. What do you think about that as a potential approach?
Yeah, no, I think there's a lot of potential there, and I think, with redosing, initially we were talking about what dose you may need years, hopefully decades down the road, but I do think it could be interesting for some of these target tissues where you may need higher doses, and if with the safety findings we're potentially seeing early on with higher doses, I think that idea of splitting that dose into multiple doses in the near term is very interesting. Obviously, after your first dose, that's when you're really going to have the highest titer of antibodies, and that's something you need to really think about if you can really get over that in its entirety or how much time you need there. But that idea of taking those high doses and splitting them up is, to me, very appealing.
Nicole, can I follow on with that? I guess theoretical, but let's say you had one serotype which you did your first dose. Could you tighter up the dose to get the efficacy you want with a different serotype, or would you still run into the antibody problem from the first one?
I think that depends on what capsid you use. So a lot of the natural variants, like AAVs 1 through 9, have a fair amount of cross-reactivity with one another. So the neutralizing antibodies against one will cross-react with many of the others. I think the hope is that with some of these newer engineered capsids, we'll see less or hopefully none of that. But I think in the short term, we're still going to be limited in the sense that if you're using AAVs 1 through 9, you're probably not going to be able to do capsid switching. Maybe AAV5 would be the exception there. But I think in the short term, we're not going to be able to do that unless we get some better engineered capsids.
Yeah, great, thanks. Nicole, can I just stick on one of the topics that came up from the FDA committee meeting was that, integration is part of AAV life now. I think probably accept that. Does that come into the way we think about dosing? Because, I always think about that non-integrated DNA tends to produce lower levels of protein than when it's linearized and actually in the genome. So how should we think about dosing if integration is part of the life of an AAV?
I think my honest answer in the short term, I don't want to say I wouldn't think about it. I think you should still be worried about the main problem at hand, the disease at heart, since most of us are working on fairly acute and often life-threatening diseases in kids. That's not to say that that shouldn't be something that should be considered at all, but I would still focus on what's going to be the best, safest, most efficacious dose for the primary effect at hand. I think the caveat or the exception there would be if you are doing high-dose intravenous administration, whether or not your target organ is liver or not, but if you're doing high-dose intravenous administration, I don't know that I would necessarily change my dose. You should still probably be motivated by safety and efficacy on the main front, but you probably want to implement some longer follow-ups, both in terms of liver ALT, AST, and ultrasounds moving forward, because you're probably going to be asked for that data anyways, so just plan for it.
But that would take time to generate. Those linearizations seem, especially in, say, the dog data from UPenn, took quite a few years from what I remember. That would really add a lot of time to our preclinical packages. Tim, what's your view on this?
Yeah, I liked that the focus was on the risk-benefit discussion, really around integration in particular. And, let's think about this for a second. So you've got Zolgensma, right? Big systemic dose. Kids 90% of them are going to die by the age of 3. They're up walking, talking 5, 6, 7 years post-dosing. Pretty sure we did our job, and I don't think that integration is really, it's never been clinically shown to be anything in this 3,500, I think, patients, right, that we've talked about. So while I think that it's important for long-term studies, we want to do long-term follow-up and find that, but again, if you're going to treat a rare disease patient, where there's nothing else out there for them, and 20 years later, there might be a non-tumorigenic tumor something that comes up from integration, I still think that we've done our job.
No, I think as an industry, we've done incredibly well with what is incredibly hard science. I agree. Yeah. And we've really focused, I believe, on safety, efficacy, and trying to manufacture. But as we move forward, and I'm aware we've only got not many minutes left of this panel, I'd like to ask each of you in turn, starting with Eric. As we move forward over the next 2, 3, 4, 5 years, what do you think we're going to be able to do to really improve this dosing topic? So when we come back here in 2026, we say, yeah, dosing, we're well on the way to solving that. What would you like to see?
Yeah, so I guess it starts with manufacturing in pharmaceutical development. Certainly, everyone's trying to focus on the quality of our products, increase the ratio of full to empty, so we're delivering as much of that transgene as possible. And I think we've all seen that manufacturing can be challenging. A lot of companies have
It is.
kind of
I agree. It's totally challenging.
have won and lost on that front. So I think that's where it starts. And then, I think, we continue to learn and we're continuing to build on those learnings. So I think, we're really understand the safety of this. And if you really think, on the totality of, if there's been over 2,500 trials with this many patients, we've learned a lot. So I think we just need to continue to build on that.
Do we share enough?
I have to tell you, I've been very impressed with the amount of collaboration that's been done across the companies. I think certainly it helps that it is a small world. A lot of this gene therapy has been done in the field of rare disease. A lot of us, we all know each other for a very long time. So I have never been turned away when I've reached out to a company to understand what they're doing or ask for help. So I would say, while that's always a concern with industry, I've been very, very impressed with the level of collaboration.
We have a fabulous industry, super friendly and super helpful. I absolutely agree on that. Peter?
Building
Crystal ball time.
building on that excellent answer, I think we are dealing with a real complex biologic, and I think in the last few years it's with the number of patients now treated, it's really made clear what the challenges are ahead for the next 5 to 10 years. And I think it goes to every aspect of that product design, from manufacturing. We've talked a lot about capsid and new approaches that are out there to modify the capsid, because ultimately it's about delivery with the capsid and lack of immunity or avoiding the immune system. So I think we've developed engineering tools. I'm expecting to see new variants appearing that are neutralizing antibody evading, but also, to Nicole's point, detargeting the liver, for example, more disease first approaches with vectors. And then I think we now understand the constraints of the payload very well. It is incumbent upon us now to work on new promoters and new transgene technologies to really maximize the expression and longevity of expression for our patients. So I'm expecting to see all of those things progress, but I think that progress is now going to be exponential. I think we really understand clearly what the challenges are and what we need to do for our patients. And so I really look forward to a very bright future. As you say, the industry is very collaborative. There's a lot of data sharing that's been going on at multiple levels, even at the disease level and foundations and things. So I think we have a bright future for AAV gene therapy technology industry-wide.
Yeah, I agree. We're in for exciting times. Tim?
Well, one of the fun things about viruses is that they're a great delivery vehicle, right? And so as we look to the next 5 to 8 years, we're seeing more and more prime editing, base editing, CRISPR coming out. So, those are going to require sometimes 2 AAVs potentially. So I think that when we look at the next couple of years, you're going to see more routes of administration using AAV with more routes of gene editing. That's going to really kind of move the field in a whole different direction. So looking forward to seeing that.
Agreed. Nicole, the final word is yours.
Final word. So most of the answers already shared are the most obvious ones, so I won't repeat those, and perhaps end on a forward-looking statement that I'm incredibly bullish about AAV. I mean, I'm an AAV gene therapy professor. I've hitched myself to this horse. I'm confident that we will not only solve the dosing and redosing strategies in the next few years and be able to not only treat patients who aren't eligible and redose patients and all of these things, but I think we'll get so good at AAV that we'll now have the very unique and interesting challenge that we'll have patients who were treated as children for perhaps a rare monogenic disease who will now grow into adulthood and who will need another gene therapy, but not for a monogenic disease, but perhaps for an acquired disorder that'll happen with, so things like diabetes, things like congestive heart failure, these types of things. And so that'll be kind of the next challenge that we get to go after, and I'm excited for that day to come.
What a great note to finish on. So I'd really like to thank our panel of Eric, Peter, Tim, and Nicole for a really fantastic session. Thank you very much indeed.